Bismillahirrahmanirrahim. Assalamu alaikum. On behalf of the Sindh Institute of Urology and Transplantation, I would like to welcome you to this special webinar to recognize and understand the scientific contributions of Professor Mohiuddin Mohammad in making animal to human organ transplantation, cross-species or xenotransplantation, an actual possibility in the future. By overcoming long-standing scientific and medical barriers, Professor Mohiuddin and his team have actually made this possible to have a animal heart beating in a human chest and sustain life, demonstrating real possibilities for animals as a source of replacement for organs for humans who need them. Dr. Mohiuddin has not only advanced science and medicine, he has also brought laurels for his alma mater, Dow Medical College, and for his country of origin, Pakistan. SIUT celebrates that. Today, Dr. Mohiuddin will be talking to us about his 30 years plus of research in cardiac xenotransplantation, how he got where he is and where the field is right now, and what's ahead for him and for his field. To engage with Professor Mohiuddin for his talk, we have a panel of 4 whom I will briefly introduce right now. So our panelists include Professor Farad Muazzam, who is the founding chairperson of the Center of Biomedical Ethics and Culture, CBEC, at SIUT, which happens to be the only bioethics center in the country and has been in active service for over 17 years now, imparting bioethics education, research, and capacity development. Professor Muazzam is also a pediatric surgeon and established the Department of Surgery at Aga Khan University in the 1980s. Our second panelist is Dr. Mirza Nafi Zafar. Professor Zafar is a professor of pathology and head of the research and publication department at SIUT. He's also heading the transplant immunology and tissue typing department, which he helped establish at SIUT over 35 years ago. Our third panelist is Dr. Aisha. Dr. Aisha is a genetist and has a PhD in genetics and has an interest in transplant-related genetic issues and will be speaking, asking questions regarding this particular area. Our fourth panelist is Dr. Wajiha. Dr. Wajiha Musharraf is an assistant professor of immunology at SIUT. Her area of interest includes transplantation and immunosuppression, which will be her focus of questions for this session. So, Professor Mohiuddin, since the day the news broke of the successful experimentation with cardiac transplantation, our director at SIUT, Professor Adil Hassan Rizvi, has been extremely keen on engaging with you. And his preference, of course, was physical presence. However, given your current work commitments, Dr. Rizvi has settled for this virtual meeting to start off with. I would now request Professor Adibul Hassan Rizvi, the director of SIUT, to say a few words of welcome to our distinguished speaker. Professor Rizvi.
It is a great honor to have you here, sir. We are honored that in spite of such a busy schedule, you have taken time off and you are giving this talk. There's a lot of things have happened during a few years, but something which has brought honor to the nation, encouragement to the younger generation is your work. And we're extremely thankful. And we pray Almighty that you should make progress further and more and more. The only request I would like to make is that you should take some time off, if possible, to visit personally to this country. And we are very keen to receive you here. And that will be great encouragement to our younger generation. We want to show that in Pakistan, there are people who are doing good things as well. We want to show to the nations of the world that we are not only bad people as pointed by the press. we have got intellectual, we have got selfless workers, we have got those who are going to change the fate of the universe by their research and their hard work. So my request will be that if you have got any plan, any visit this direction, please let us know. I would like to receive you.
Thank you. Thank you very much, and I will. Thanks.
Thank you, Professor Rizvi. Moving finally towards our distinguished speaker and his formal introduction, I can think of no better person for this task than his friend for over 40 years, Dr. Munir Amanullah. Dr. Amanullah is a pediatric cardiac surgeon, a rare breed in this country and a trailblazer in his own right, and is currently working at the Liaquat National Hospital in Karachi. So, Dr. Amanullah, over to you for a formal introduction of Professor Mohiuddin.
Thank you, Dr. Jafri, or should I call you Amir? But what an honor and a privilege and a pleasure to introduce Professor Mohammad Mansoor Mohiuddin. Dr. Mohiuddin is currently Professor of Surgery and Director of Xenotransplantation at the University of Maryland. Prior to this position, Dr. Mansoor was Chief of Cardiothoracic Xenotransplant Research at the National Institute of Health in Baltimore. Dr. Mansoor's journey has taken 30 years in xenotransplantation to achieve the impossible. But Mansoor's journey did not start in 1992. It started with Mohiuddin and Abida being blessed with their eldest son, Mansoor. Let me introduce the man, Mansoor, whom I have known for more than 40 years. And it will be fair to say that we are chuddy buddies. Mansoor did his schooling from PAF Model School, Faisal Road, where he was a good student and an outstanding athlete. He was friends with two legends, Junaid Jamshed and Tazeen, my wife. PAF under Principal Simon DeMella was highly disciplined, and that's what Mansoor has got in his genes. Mansoor's other strength is his passion. With Mansoor, it's all or nothing. Quite a few of us would remember during the DMC second year, we would have the season of making clinical groups. We had identified hours, we approached, they refused, we were devastated. To overcome this sorrow, Mansoor drank Benadryl and went to sleep. But he was back the next day in high spirits. This is Mansoor, a never dies attitude. Mansoor continued to do well in medical college, but he excelled in athletics and was Karachi champion in long jump and triple jump. He also led the Karachi universities to third position, which they hadn't had for a long time, at the All Pakistan University Athletic Championship. He would train so hard that we had to do as well. He inspired us. After doing his MBBS, he relocated to USA and started his career as a junior researcher in Philadelphia, and then progressed to senior researcher and now director of Xenotransplant. What Mansoor has achieved today has been due to sheer hard work, passion, discipline, and self-belief. Throughout his journey, he has been supported by Nida, his wife, who is an artist, and his children, Hiba and Hamza. Mansoor is currently president of International Xenotransplantation Association, president of Dogana, which is the USA-led Dow Medical College Association, associate editor of the Journal of Transplantation in Frontiers, and as director of Xenotransplant, he has a grant of $4 million every year. He's very well published, respected, and as a scientist. He and his team holds the world record of the longest surviving xenotransplanted animal of nearly 1,000 days. Mansoor has achieved what many thought was just a dream. This he has managed by his persistence and having the self-belief that it's possible. Mansoor and I have had many long discussions about xenotransplantations over the year and never had any doubts that he would not do it. That's because he is Mansoor. Before I end, I just want to remind Mansoor that from now on, you are my best friend. My suitcase is packed for the flight to Stockholm. Mansoor, congratulations, and thank you for making us all proud Pakistanis. Ladies and gentlemen, I present to you Professor Mohiuddin, most probably a Nobel laureate in the future, inshallah. Mansoor, over.
Thank you, Munir, and thank you, Dr. Prisby. It's indeed a great honor for me to be here. When Dr. Prisby asked me to join, there was no question of me saying no. I have such a great respect for Dr. Prisby, and he's one of my idols, and I'm sure idol of many of the people sitting here. So we look up to his achievements, and whatever I've done, it's because of role models like Dr. Prisby. If I could share my slides, I will. Do you guys see one slide or two slides? Do you guys see my slides?
Yeah,
Yeah.
we can see your main slide and also your the slide sorters are on the right. So you.
Okay, so I have to swap it then.
Yeah.
Now you see only one?
No.
Yes,
Yeah.
we see only one.
Okay, good. So, so thank you again for this opportunity. I'll take you back not 30 years, but at least 15 years of research that led to this transplant. It's been like a roller coaster journey, and as you know, the research is never rewarding all the time. There've been times that there was time of despair and frustrations, but there have been times where Allah subhanahu wa ta'ala has rewarded me tremendously. So this is a long journey. What we have achieved now, honestly, I never imagined that we could have done it at least in my lifetime, because of a lot of negativity surrounding this field, and the progress that was very slow, and all the experiments have been very, very expensive. So as you guys know, the reason we are doing xenotransplantation is because there's a large number of patients now waiting for an organ transplant, and no matter what we do, how much awareness we will create, this list will not go down, and most of these patients die waiting for the transplant, as shown here. And this is data from United States, and you can multiply it by several hundred folds when we talk about the entire world. So based on this data, one person every 80 minutes is dying just waiting for this transplant. So the one option for this is xenotransplantation, which is not new. It has been tried for the first time in 1682 when a nobleman lost his piece of skull, and a piece of dog's skull was transplanted to save his skull, and that was done successfully. Later on, several burn patients received frog skin to repair their burnt skin. But in the recent years, if you remember, and some of us may remember this, the baby heart was the most famous in 1984 when this heart was transplanted by Leonard Bailey in Loma Linda, and this heart survived for 3 weeks. This is perhaps the longest xenograft ever survived, and this heart came from a baboon. Along with Leonard Bailey's, Keith Reemstema did kidney transplants in 1963, even before I was born. And James Hardy also did chimpanzee heart in 1964. In the recent times, Tom Strazel did a liver transplant, but none of these survived for a long period of time, because most of these were done without long research and without proper data. Just it was a last ditch effort to save the patient's life. So I mean, we suggested that we should use pigs instead of baboons for several reasons. I mean, when I see I refer to a large number of people who have been working in this field for even longer time than I am. So the pigs were chosen because of their ability to genetically modify them. That we know the genome very well now. We have techniques to modify their genes. They are anatomically similar. Their organs are anatomically similar to humans and baboons, which we used as the recipients in our preclinical studies. They are farm animals. They breed well in captivity. They have a quick breeding cycle that required size within months. And in large part of this world, they are consumed in food as food, and there are less ethical issues because of that. So, and their physiology, as we are learning, is very similar. There is some differences in the heart rate and few other things, but overall, the physiology is very similar. So what happens if you take a pig heart and transplant it? In this case, in my initial studies were to transplant them in the abdomen, with leaving the original baboon heart and just transplanting this heart in the abdomen to see the effect of various genetic modifications and also different immunosuppression. So when you transplant an unmodified heart and without any immunosuppression, this is what happens. So within minutes, the heart looks like this, and it rejects very quickly with what we call hyperacute rejection. And what we found in the vessels is like there was microvascular thrombosis that happens very rapidly and causes it to reject. And the other findings include, loss of myocytes and bleeding within the interstitial spaces. So what happens? One of my colleagues, Uri Galili, several years ago discovered that millions of years ago, the old world monkeys, apes, and us have lost this enzyme. Let me just get the, so if you can see my cursor. So lost this enzyme due to some calamity. And because of that, we stopped having this sugar on our endothelial cells, which lines the blood vessels. Because of that, we develop antibodies against these antigens. And when we transplant this baboon heart, baboon organ, and the vascular system comes in contact with these antibodies, it causes the endothelial cells to swell up, expose lamina propria, and then causes rapid coagulation and thrombosis of the vessel, and then leading to the death of this organ. So, I mean, over years, we started to find different reasons for these for the rejection, and I describe it as the process as an onion. As we started peeling off one layer, we started discovering other issues, and all these years, our focus was to get to a stage where we can comfortably say that we have dealt with major issues, if not all. So in my research, my first success came in. So until 2001, the survival was very difficult. In 2001, if you remember, just before 2001, they discovered this technique of cloning, and the first dolly sheep was made, and the same company then knocked out the carbohydrate that I showed and the enzyme from the pig cells, so that don't express that carbohydrate, and therefore the antibodies we produce do not cause rejection. So with that, using that animal and another gene, CD46 transgene, we for the first time showed that a long-term survival about 230 days was achieved. This was also achieved by using the immunosuppressive regimen, which I'll talk a little bit in detail later on, requires elimination of B cells with rituxan and using a co-stimulation blockade antibody. In this case, we used anti-CD154, which is also called CD40 ligand, and we got survival. Until this time, the longest survival used to be 180 days. We got about 250 days. So that was the first success. But later on, we got about like a year survival, which was considered a milestone. And then we found out that this anti-CD40 antibody that we were using was causing its own complication and was causing thrombosis in the animal. So by itself, because it is expressed on platelet and causes platelet aggregation. So with that, we switched to CD40, which is the same, which is the antibody that blocks the same co-stimulation pathway, but on the other end. But when we use the same dose of that antibody, we were not able to survive, to get the long-term survival as described here as in the green. However, when we doubled the dose of that antibody, which we later found out that was due to the instability of this antibody, we were able to get survival for a long period of time, as shown in red. So throughout my career, I've kept my regimen the same, with which I'll describe in a bit. And with this regimen, we were constantly now getting long-term survival beyond years. So this is the regimen that we finally started using, and a lot of people call it Mohiuddin's regimen, but I have not called it Mohiuddin regimen yet. So this includes induction with a couple of doses of antithymocyte globulin to remove T cells, and 4 doses of rituxan over a period of 3 weeks, and also we use different agents to block complement, either the cobra-1 factor or recently C1 esterase inhibitor. Our maintenance is predominantly with anti-CD40 antibody and MMF, and we taper down the steroids and stop them within 2 months. We give some supportive treatment. This is all baboon work. We give heparin continuously throughout the period of survival, and to prevent CMV, we give ganciclovir. So this is the mechanism that we targeted the most. So as you know, that antigen presenting cells express CD40, and then the T cell express CD40 ligand. Our goal is to stop or prevent this interaction. But in xenotransplant, as the main culprit is the antibody-mediated rejection, our main target is stopping the B cell activation and also stopping its help to the T cell. And in that way, we are stopping T cell activation also. So both these pathways that I'm showing are blocked by either CD40 or CD40 ligand, which I've used before. So this was one of our seminal papers that showed that we can get long-term survival of cardiac xenograft in the abdomen, but when we started to decrease the dose of this anti-CD40, still we got the good survival until we, thinking of tolerance, we completely removed this antibody, and we found out that these hearts rejected as soon in a couple of months after removing this antibody, when all this antibody was washed out of the system. So this histology is just before around like 2.5 year time period. This is a biopsy specimen. It shows that it didn't have any signs of rejection. So this was like our proof of concept that with this model we established that we can avoid xenograft rejection. We have a genetic platform that we can use for the pigs, and we have an immunosuppressive regimen that is effectively working in a xenotransplant model. So this is just to show that how the survival went. I started my research in 1991 until we started. I started at NIH, the large animal model, in around 2005. Until that time, the survival was very little, less than 100, most of the time 1 or 2 going over 100. But since we started, we slowly, all this work is my work in our lab. We've slowly showed progress of survival, and finally the longest survival ever for up to 3 years. So now the logical next step was to transition and to see if this heart that is not rejected in the abdomen could support life. And for that purpose, we transitioned from an abdominal model to an original orthotopic model, where we took out the baboon's heart and transplanted the pig heart. Unfortunately, that transition was not smooth as anything in research. Any change is not always translatable. So our initial work had a lot of hurdles. All the hearts within 48 hours started to fail. But on histology, they don't look like that they were rejected, but there were some other mechanism that they started to just run out of gas, basically. So while we were doing all these transplants, we also continue to manipulate the genetics system of the pig. And this is not done at our lab. It's done with in collaboration with a company called Revivicor, which is a subsidiary of United Therapeutics. This is the same company that cloned Dolly sheep in England and known to be PPL when it was acquired by another company here in the United States. It was called Revivicor. So just for people who are not familiar with how these genetic modifications are done, here you can see that what is done is you take out the eggs from the ovary, from the ovary, and then you take out you suck out the genetic material from those eggs. And then you have a construct of the desired genetic material developed outside the body, and then you insert that genetic material into the eggs, and then put the eggs back into the ovary. And after like 114 days of gestation, new pigs are born, which have the new genetic material. Then the next step is to see whether the genes you desired are expressed or are deleted. So on the left panel, the flow cytometry is showing the blue. The pink ones are in a naive pig versus the blue one is showing that all these 3 genes were deleted. And then you see the expression of the human transgenes that we express, and you can see it with the Western blot in the column D, and also you can see on immunohistochemistry. So this is just to verify that what we wanted is in there. So these are some of the genes that we have modified. We found out that there are like 3 carbohydrate genes that are immunogenic to humans. So we have knocked them out. As you may know, that the pigs grow very fast. So there was a concern, and some evidence that the heart or other organs, especially kidney, that can grow fast in another species also. So for that matter, we knocked out this growth hormone receptor gene to control the growth. So to make it the pig still grows, but at a very slow rate. Then we focused on several other genes. Some of them, the HO1 is an anti-inflammatory gene. The HLA-E is also a kind of anti-inflammatory. We put 2 transgenes to suppress the complement activity, CD46 and CD55. Then we also found out that there's some incompatibility between the human and pig coagulation system. So we put a couple of human anticoagulatory genes like EPCR and TBM. So as I mentioned, our initial success was very limited, and all these hearts start to fail within 48 hours. At that time, one of our German colleagues in Munich discovered that this solution, which is at that time called Steen solution, which has all the electrolytes, hormones, and other inotropes, and on top of that has cocaine in it, is a good preservation solution. And if you preserve the heart with this solution, you can overcome that 48-hour failure, which we termed as perioperative cardiac xenograft dysfunction. So the heart is taken out, put in this machine that we are the only one who have this machine in the United States, but it's used clinically in Europe. And you preserve the heart for an hour to 90 minutes with continuous perfusion with this solution that also include blood, and then you transplant the heart, and then we have not seen any failure from that point on. So this is just a heart, if you guys could see, just after transplantation in the chest, and it functions very well and continues to function for a long period of time. So this is some of the recent results. As you can see, each step, it was very progressive from orange to purple to blue to green and finally red. So there was a progress in survival based on various things, various genetic modifications, as you can see. So that the pigs that we are using right now have 10 gene modifications, the same gene that is used in Mr. Bennett in human, which has 4 knockouts and 6 human transgenes. And we have seen progressive improvement of of graft function and longevity after these modifications. Although we have kept our immune suppression exactly the same as I described. So there was no change in immunosuppression, and and we got survival based on different gene modification. So, I mean, this was a famous quote of Thomas Stasel, which kept me going, that, history tells us that procedures that were inconceivable yesterday are barely achievable today and often became routine tomorrow. So that was my goal, to see the xenotransplantation becomes a goal. This is my team, the lab team. So the story doesn't end here. So this is when we had like a 9-month survival of an orthotopic heart. So we just had some celebration of that. So then, we took these results to the regulatory agencies here, FDA, and say, is it good enough to start clinical trials? They said, no, it is not. You need to do some more non-human primate work, have some kind of consistency. We need you to do 10 more transplants with 6 of them surviving for 6 months. And then we will think of giving you the permission for clinical trials. But at the same time, we found out about a opportunity where FDA in special circumstances allows use of an experimental drug to save the life of someone who has no other alternatives left. So our cardiac failure team identified a patient, Mr. Bennett, who had no other alternatives. So for his non-compliance, he was not a candidate for human transplant. He had repeated heart arrhythmias that disqualified him for the use of ventricular assist devices. And so we also checked different hospitals to see if anyone else would take him as a heart transplant patient, but none of the hospitals, agreed to that. So this is a picture that showed that, we started preparing a proposal for FDA. It's not just like a 2-page proposal, to ask them that, can we use this experimental procedure to save the life of this patient who probably would have, died or had to be, kept on ECMO support, which he was on for the last 40 days for a longer period of time. But there was no hope for him. So we submitted this application, and the FDA, and this was December 20th, and as you know, this is holiday period in the United States. So I mean, the FDA responded to us and asked for more details. So we went back and forth multiple times. So this is a board in my office, which I have not erased yet. It shows you that, the timeline on the right side, that, how many times we went back and forth with the FDA. On the left, it shows, how many permissions we had to get from the institution. So it was not only that FDA, but even the institution, we had to take multiple permissions and had to submit a lot of paperwork. So and the major barrier after, when we got permission on the New Year's Eve, but then the main issue was who will pay for this transplant, because so far it's been a month. We have already spent $1 million on this patient, and this will not come out of any insurance. So the hospital had to absorb this cost, and that was a difficult decision for the hospital. in the time of COVID, where a lot of resources are being diverted to COVID patients. But fortunately, they finally came up with the money and they allowed us to do this transplant. So these are some of the pictures of transplant. This is on the top right, this is my staff where we harvested the pig heart. The top picture is where the pig heart is harvested. It was put in this machine. It was still, this is the heart that was transplanted. You must have seen some pictures in the press, and it was finally transplanted and into Mr. Bennett, as you can see here. And you can see the function of the heart just after taking the cross clamp off. So this is so what we saw in this Mr. Bennett's heart when we took the Mr. Bennett's heart out, it was dilated several folds, more than we expected. So on the left is Mr. Bennett's heart, and on the right is the one after transplantation, the pig heart. So you can see that there was a huge disparity. The pig heart was smaller, so we had to modify the anastomosis of the heart. We have to make a smaller cuff of biatrial anastomosis in Mr. Bennett to accommodate the pig hearts shown here. So this was function on day 0. There were people who can appreciate it. Certainly Munir can. The function seems great. This patient stayed on ECMO for a couple of days because we slowly wanted to transfer the load on this heart. And after 4 days, we took the ECMO cannulas out, and this heart was supporting the rhythm. So here you can see that the same function at different time points, and the heart function continues to be very well. That was the short axis, that's like a cross section, and this is long axis showing the function of the heart. So we looked at the strain analysis, which is one of the tests to see if the overall function of the ventricles are good, and they are equally contracting. The more red it is, the better the function is. So as you can see, a lot of red in the contractions. This for day 0, and this is like day 14. Unfortunately, today is day 29 of this patient, and so we have reached almost like a month after transplantation, and his heart function continues to be the same. We have not seen any signs of rejection. The one thing I would tell you that this patient was severely deconditioned. He was on ECMO for last 40 days, and he had not gotten out of bed for last 2 months before transplant. So all his muscles were severely wasted, and he could not even sit by himself. So the major challenge that we have now is not the heart. I'm very confident, based on all the research we have done, that this heart will not reject immediately. But because of the patient's condition, we had to reduce our immunosuppression significantly, because even at the time of transplant, his WBC count was very low. So we cannot have given the same amount of anti-thymocyte globulin to him, same amount of Rituxan to him. So we had to reduce the dosage. But one thing that I did not reduce was our anti-CD40, which I firmly believe is a major player in graft survival. So our main challenge now is not to keep this graft from rejection, where we are confident that it will continue to function well. Our main challenge is to fight infections in this patient because of immunosuppression and other areas. His renal function due to this surgery was depressed, so it's slowly coming back. So we have to support him with dialysis a little bit, but it will be a long haul. I mean, he will be in the ICU for a long time, and then there will go through long rehab before he will be discharged, hopefully to his home. So, I mean, this is just to show you that what we are monitoring in terms of immune surveillance and monitoring rejection. I showed you echocardiography, but besides that, we are looking at, immune mechanisms of rejection. There are different tests. The AlloSure and AlloMap are new genetical gene profiling markers that are being introduced into this system, which have not been used before in this kind of experiments. But we are using troponin, which is a reliable marker of rejection, and we are looking at different antibodies, against the pig cells. So we think that we had learned a lot in a non-human primate model, but in a previous experiences where we moved from small animal to large animal model and had a lot of surprises, we think that there will be few surprises in humans also, because though it's close to baboon, but it's not the same. There are a lot of differences, and we expect them. One major concern that FDA had, and some of you may know of, is the transmission of viruses across species, and we have, I mean, although there is no evidence of any disease caused by these viruses that have shown to have infected human cells in petri dishes. One of them is the porcine endogenous retrovirus. This has shown to infect the human cell lines, but there have been some studies done in New Zealand where islet transplantation has been done from pig to human, and long-term monitoring of these patients shows that they don't have these kind of any disease, any porcine disease. But anyways, FDA was very strict, and their major concern was to monitor these kind of infections. So the patient had to sign a consent that he'll continue to be monitored throughout his life for these viruses. Not only the patient, anybody coming in contact with him had to sign a consent that they will if this patient develops some kind of disease, they will volunteer and come back and have them checked for the disease. So I mean, that this was the assurance we had to provide to FDA to get this permission. So where to go from here? So, and then the next big thing will be discharging Mr. Bennett from the hospital if everything goes well and he recovers well, which still, I would say, about 2 months away, because he will require a lot of rehab. He started his rehab a little bit. When we started, he couldn't even hold his neck up, but now he's able to hold his neck up and and sit properly. He wishes to go to the bathroom by himself, but he's not fit enough to do that by himself. One thing I didn't mention, then when we took the consent from him, which was a big thing, because it's a 4-page consent with everything possible explained to him. We never promised him a long-term survival. We told him that he could not. There's a possibility he could not even make it through the surgery. But he realized all that. We had 4 psychiatric consults done, 2 internal and 2 external, to determine his ability to give that consent. And then the ethics board reviewed those consent and you approved. So it was a long process just to establish that he's mentally capable enough to give the consent to this. And he told us, just to let you know that he was mentally capable. He said that in response to this, when we asked for his consent, that okay, once I have this heart, will I do oink oink? So he was, and now, I mean, he's pretty much in a condition that, he's a big football team Steelers fan. So he asked about how they are doing, and he knew that one of their quarterbacks retired. So I mean, he's mentally still doing very well. So it'll take like a while for him to discharge. But the FDA will not take this as an excuse for us to go and start doing this on a routine basis. They still want us to fulfill all the requirements of non-human primate studies and go through their proper route. There are other institutions after us who are desperately trying to, and several of them in kidneys, to get similar permissions, but none of them have the data that we have collected over years. So it remains to be seen that who else can get the permission to do this one-off. But we require other institutions to participate, because just with one human experiments, we cannot generate the data that will convince the FDA to let us keep us in this process and let us do more more experiments like this. So this is a couple of pictures. This on the left, this is the first week after the transplant. You can see the patient is still, is reasonably attentive. And the next one is just last week when the other person in that picture is Dave Ayers of Revivicor, who was instrumental in genetically modifying these pigs. So, this patient was, talking. He has some difficulty talking because of, repeated intubations and stuff. But he's getting his voice back. On the left, the other person is his son. He lives alone. His son lives several miles away in a different city, so he doesn't come here very often. He just visited him for 2 days. So, I mean, this is something that's not available or not in the culture of this country where I live. That he's been in the hospital for several days, but had no visitors from the family. So this was the only time that his son came in. With that, this is just a. In my conference room, I made this to honor everyone who participated on this on the day of the transplant. As you can imagine, it was like a big show, and a lot of people participated. Just in the OR doing this transplant, every surgeon who had anything to do with this transplant was present in the OR. So it's a big thank to a large number of people who made it possible. With that, you've always heard this saying that xenotransplantation is the future and always will be. My goal is to change it to saying that xenotransplantation is present and it always will be. With that, I thank you very much for your kind attention, and I'm happy to take any questions.
Fabulous walk through your fascinating journey, Dr. Mohiuddin, of 30, if not at least 30, 15 years of the past, in which you've actually made the future the present. So that's actually a very, very significant contribution that you've made towards medical science. So I would like to now ask our panelists to put some questions. The first, I'd call upon Dr. Naki Zafar to put his questions to you. Your floor is yours, Dr. Zafar.
First of all, unbelievable achievement. You have summarized centuries into 15 years by achieving this really game-changing research. Whenever we talk of xenotransplant, there are always two questions. Some of them you've already answered. You may not have data for survival of Mr. Bennett, but from your animal studies, what does your data suggest? How long would this transplant survive in human? And my second question, which is always a concern, and you probably will have problems with FDA, is the transmission of disease. So you have any evidence from your baboon research, pig to baboon, of this disease transmission? And what kind of surveillance will you do, or you're planning to do in your research when you do go ahead and you get permission to do some of these transplants again in humans?
Yes, so I mean, I'll start with your second question first. So as I mentioned that it has always been a major concern. The safety is the major concern of FDA in any new experiment or drug. In this particular case, we asked for not only for permission for the pig. You know, the hard box that I showed is also not FDA approved, and the drug that I'm using, the anti-CD40, is not that approved. So all those three things were to be approved together. In terms of surveillance, and the major questions was from FDA to develop a surveillance plan to show that, you know, there is first that the pig doesn't carry or any disease, and then to see, you know, to follow this patient through and see if he develops any kind of disease. So you know, when we check the pig, the virus we are more concerned about is the PERV virus, porcine endogenous retrovirus. This virus has three types, A, B, and C. So this pig did have, and most pigs have this this endogenous virus. This pig had porcine A and B. So our work will be to demonstrate that at any time point, this patient doesn't have this virus incorporated into his genome, or we cannot detect this virus in this patient. So far we have detected, we have tested for this virus until day 29. You cannot do this test every day. You know, you get the RBCs, or the peripheral blood lymphocytes, and do PCR to see if this virus is present. And so far we have not seen this virus. And this test will be continued to be done throughout his survival. And if any time point, you know, we see this virus being transmitted, not only him, but anyone who came in contact with him, and especially intimate contact with him, will be evaluated for the transmission. So that kind of assurance we had to provide. What was your first question? I'm sorry, I missed that.
The first question was from your data on pig to baboon. Do you have any timeline, or can you predict survival of the transplant in humans, or Mr. Bennett for that matter? Is there a timeline?
Yeah, so I mean, you know, every day is a bonus for us. I mean, you know, our first goal was to see if this heart undergoes hyperacute rejection within minutes or hours. That did not happen. So that was the first victory. Then we also, as I mentioned, you know, initially our hearts were failing within 48 hours without rejection. So that did not happen. So that's another victory. So our longest heart in its original position, chest, survived for 9 months in a baboon, and that baboon died with some other you know, infectious complication. But so when we took that heart out, it did not have any histological evidence of rejection. So we are hopeful, you know, we should get good survival of the heart. But as you can imagine, all the baboon work was done in healthy baboons. So, but this patient is a lot different. You know, he's severely debilitated. Although all the other organs are working fine at the time of transplant, but you know, he's not a normal human being. So we have to carefully adjust our immunosuppression. Continuously, we are fighting infections because you have to create a balance between immunosuppression and prevention of infection. So that's the biggest challenge. So which we have not faced in our baboon model. So we are taking every day as a new day, you know, and it's hard to predict, but we at least hope that, you know, he will survive for a significant period of time. If for any other reasons, you know, he suffers. So let's see. I mean, you know, we've gone. We have already at one month mark. So I count first, you know, hours, you know, days, weeks, and months, and then years. So we are on the 4th stage already.
Well, I hope you continue on the months.
Okay,
Thank you.
Thank you.
So a lot of genetic intervention, of course, went into preparing this animal for a successful heart transplant, the pig to the human. And Dr. Aisha, who has an interesting background in genetics, I think you have a few questions regarding this.
Before that, Dr. Jaffrey, I think Dr. Rizvi, Dr. Adib Sahab wanted to ask
sorry,
something. Yeah, I think he was trying to ask something.
Achha, sorry, go ahead, sir.
Just to tell you how proud we are, and can there be any possibility of opening an experimental lab in Pakistan?
So the major thing is the cost. So each transplant we did, and it survived for 6 months in an orthotopic position. Besides the cost of the pig, which was given to us for free because the sponsor is our collaborator, it cost about $500,000 per transplant. So I mean, you know, so the cost is the major factor. Then the other factor is the availability of these genetically modified pigs. To my knowledge, at least in the United States, there are only 3 companies who are making it. I don't know if these and in China they are making it also, but you cannot transport pigs across to different countries. So I mean, for this kind of research to be done in Pakistan, the first thing that will be required is to get those pigs somehow, either from here or from China. I mean, when this company started making it, they told us that, you know, initially it was costing them $500,000 to make a one pig. So, but the cost has gone down remarkably because now with CRISPR-Cas, Cas9 techniques, they don't have to breed them to get these multiple gene knockouts or transgenes. they can do it in one shot. So the cost has significantly dropped down. But whether or not this can be done in Pakistan, it all depends on, if this facility is available to genetically modify these pigs. Of course, I mean, anything can be done anywhere. The cost and the availability of facility will be the limiting factor.
Thank you.
And now moving towards Dr. Aisha, you have some questions?
Yeah, sure. Thank you, Dr. Ahmed. First of all, I will congratulate Dr. Mohiuddin for such a great achievement. And as a genetic student, I have special interest in those experiments where you've done the genetic modification. You showed us a historical journey where you performed experiments with taking 3 gene and you achieved this particular experiment with 10 genes, with the modification of 10 genes. As I understand, the main difference, the key difference between the previous experiment of Perioperative of heart transplant and your Mr. Bennett's transplant is the genetic modification. I guess keeping in mind that you maintain the immune suppression constant in your previous experiments. So based on your current data and the previous experiments that you are, and the current data that you are gathering, and you said you are making genetic profiling of Mr. Bennett's DNA, do you think there is a room for improvement in the combination of these these 10 genes that you use? Do you think in the future experiment you can have a different gene for make your pig more humanized in your future experiments?
Yes. So thank you for your question. So first of all, the baby-fay heart was a baboon heart, and it was not genetically modified at all. And the immunosuppression was not, as much, the different options were not available then. Your question is very valid, that now that we have a technique to alter the genes, what is the limit, right? We can continue to do this until we basically humanize the pig. We don't have any intention of doing that. Our main intention would be to get to a stage that, we can minimize the immunosuppression to very limited amount, or completely avoid it in case of if we are able to induce some kind of tolerance. There are other companies who are also doing this kind of work, making the pigs, and they have done far more genetic alterations than 10 genes also. But with every gene, alteration, when you are taking something out or modifying it, you're compromising some of the functions of those genes in the pig. And I can tell you with our experience, there's the attrition rate of these pigs have increased with these modifications. So some of the genes with overexpression have caused these pigs to bleed spontaneously also. So we have to be careful, just because we have a technique to alter these genes, we cannot just get carried away and alter all the genes that we think are responsible for any immune reaction. So we have to create a balance. I think that the techniques of tolerance may come into play, and by simultaneously transplanting thymus with it, there are groups who are doing it, or inducing microchimerism. Those kind of techniques, along with limited gene modifications, will be very beneficial.
So does that mean that the same model works for other organ transplants like kidneys?
So you must have heard of one kidney transplant in a brain dead patient at the University of Alabama. So that was our initial goal, to first do a transplant in a brain dead, where a person who was pronounced brain dead and is still on ventilator, and if he gets permission, his family gives permission to use his or her body for experimental purpose, then we could have transplanted the heart and and looked at the hyperacute rejection for a few days, up to 2 days. So the University of Alabama and New York University have done those kind of experiments in kidneys. In NYU, the kidney was transplanted outside the body by Bob Montgomery, and they kept that kidney for 2 days and saw that the kidney produced urine and cleared the creatinine. The similar thing was done at UAB when they put the kidneys in its original position, although those kidneys did not perform that well. I mean, they produced urine but did not clear the creatinine. So the group in University of Alabama used the same pig from the same source, a 10-gene pigs, whereas at NYU they used only 1-gene modified pig with the thymus transplant. So people will try different combinations, but it appears that depending on the organ, this 10-gene will be sufficient to get some kind of survival.
Thank you very much for that intervention, Dr. Aisha. Now, clearly this is a multidimensional challenge with gene knockout on one side, and of course patient selection and ethical clearances, etc. And immune modulation comes out as a very, very important aspect, which raises its own question. You mentioned also the Mohiuddin solution, which is fascinating. Dr. Wajiha, I would like to invite you now to pose some questions to Dr. Mohiuddin regarding this area.
Yeah, greetings, Dr. Mohiuddin,
Thank you.
congratulations on such great work. You've already talked about tolerance. I basically come from immunology, so still I'm stuck there. So I would like to ask you about autoimmunity. Have you seen any signs of autoimmunity in these pigs, or have you checked this patient for autoimmunity or any markers of autoimmunity?
So there is a concern that there is some autoimmune mechanism that will come into play, although we have not seen that in our baboons, and as I mentioned, those are healthy baboons, so that's why probably the reason we have not seen it. But in Mr. Bennett, we are seeing that there's some kind of destruction of his red blood cells and white blood cells is going on. We think that could be due to some of the autoantibodies generated either against one of our immunosuppressive drugs or the broad spectrum antibiotics we are giving. So I mean, that definitely that cannot be ruled out and will be a factor that can only be studied in patients like these who are kind of immunosuppressed and also very weak and compared to our baboons.
Yeah, that's the concern. Thank you.
Thank you, Wajiha, for that intervention. Now we've been talking about the science. Okay, I think Dr. Prisby or Dr. Naki Zafar have a question. Yes, please go ahead.
Yes, I'm talking in particular for this patient because he's so heavily immunosuppressed. Are you giving any prophylactic, for example, immunoglobulins to control infections? Because in situations like this, when the patient is heavily immunosuppressed, we also give IVIG, for example, to prevent. Are you doing anything like that with this patient?
Yeah, so I mean, basically at this time, this patient is, I mean, we have already, decreased the level of MMF to a very low level. So basically he's getting only the anti-CD40 antibody and tapering doses of the steroids. You may not know that, but this anti-CD40 antibody is also an IgG molecule, IgG4 molecule. So I mean, we have not used IVIG in our baboons or in patients like this that to fight that infection, but we believe that the infection we have this carriers test that we can do to check the the pathogens. So far there were a couple of instances where we could identify some kind of infection, but so far it seems like that the broad spectrum antibiotics are controlling all the infections that could happen. Not only bacterial, we have coverage for fungal and CMV with the ganciclovir. Yeah, I mean, at this time, we have not included IVIG or any other immunoglobulins for that purpose.
Thank you.
So, Dr. Mohiuddin, while xenotransplantation is obviously surrounded by these major scientific challenges, ever increasing and very complex. However, there is also the social, cultural, religious, and ethical factors that you've already been talked about, alluded to. But to bring us into this direction, I would request Professor Farhat Moazzam to direct some questions towards you.
Thank you, Dr. Amir. Dr. Mohiuddin, I really enjoyed your talk. So you must be aware, Klaus Hammer, who's a scientist, made a very interesting comment when he was talking about xenotransplantation. And what he said was that in order for xenotransplantation to succeed, his quote was, "We will need to outwit the 180 million years of evolution." And learning from Dr. Munir that you were an athlete and you were an athlete in long jumps, I'd like to congratulate you. It seems you made an intellectual long jump in this area. And for that, of course, you need to be congratulated. The thing that I enjoyed in your talk was that within your talk, you did raise, although your focus was on the scientific areas, but you highlighted periodically the other social and ethical issues that are informed, that are part of this. So in this particular area, for example, currently, right now, there are no guidelines, there are no policies for xenotransplantation. So I was involved with the WHO guidelines that came up with for organ transplantations, and when we had reviewed that, but they're clearly out of date right now. So a couple of areas in which, perhaps you can give us your views. So as you probably know, Hastings Center, which is one of the primary bioethics center in the States, and NIH is actually funding a 4-year study in which a young lady, Karen Maschke, who's the principal investigator. And so they are beginning to look now at ethical and policy guidance for xenotransplantation and clinical trials for that. And so I'd like your views, number one, on the difficulties that are going to show up, because in order for xenotransplantation to be offered as one of the things for those who need organs, the issues of, and you've referred to some of them, perhaps you can talk a little more about it, the issues of equitable access, for example, And you've already pointed to the cost that is involved even in the United States, and also the issue of commercialization. So what are your views on this area? Because in order where money comes in, then the governments cannot give you that kind of money. So you have to turn to that. So maybe some views on that. Also, the other thing is the eligibility criteria when you're moving to clinical trials. Right now, the permission you have gotten is for one time, and possibly FDA may give you a chance to do some more. So one on how do you see, I would think that there is a need for policies and guidelines. And the other thing, which so some views, comments, do you see an issue with this moving towards clinical trials? So that's one area. The second area, and you also alluded to this, Nuffield Council on Bioethics from UK has also been, everybody's weighing in, and they talked about the surveillance issue. So the issue there is, in Mr. Bennett's case, he's relatively isolated. He's, in the hospital. He doesn't have a large family. So the second area that I would like your views on is what happens when you do this, and there is family contacts. So the surveillance for zoonotic kind of infections, including also to the healthcare providers who will be following them. To me, that seems like a kind of a major issue that will need to be looked at. So in the informed consent, somebody with a large family, would the informed consent of this also involve then an informed consent of the family members that they will be willing to be surveyed for the rest of their lives? And the other issue, what if the patient says in a clinical trial, that's it, I've had it, I want to get out of the trial, which would be an interesting situation. So perhaps you could talk a little bit about the clinical trials, how you see the difficulties with it. Thank you.
Thank you very much. As Munir mentioned, I'm president-elect for the International Xenotransplantation Association also. So we started thinking about this several years ago, and so we have this communique out. It's called Changsha communique, which we met twice in China 4 years apart, and developed some guidelines for and we involved WHO in it also, to address some of the ethical issues. And whatever you have described are valid issues, and we have faced some of them already. In terms of the surveillance, we have the patient had to sign a consent that he will inform us of all his intimate contacts, and we have every single person who's coming in contact with him has to give a sample. Some of the ones who are coming in frequent contacts, like myself, have to give out a blood sample that will be kept for a long period of time, just to make sure that we didn't get any viral transmission. This person, though now we have only one patient, that it will be easy, and we have promised that we will keep a close eye on him even after he's discharged, because one of the reasons he was not offered a human heart was his compliance. A major question that came up was, how do you know that he will be compliant with this heart, and takes his medicine and all. So, but we have promised the FDA that we will follow him and make sure that he does that as long as he lives. So, but that will not be possible for once there's a bigger clinical trial. There are not resources enough. So, those are the very valid questions, and they will come into play once we started enrolling more patients. And definitely, FDA will be interested in developing a more thorough plan for multiple patients, which will be more practical, right? So you cannot follow every single patient and their intimate contacts for a while. So this patient also signed that, if he comes in contact with anyone, he will inform us. But you cannot, rely on that. So these are ethical issues that we have already encountered in one patient, and they'll be multiplied once we have. So yeah, I mean, I think we'll have to deal with it as we go along. And there'll be many issues. I usually don't read the comments after on the social media. A lot of these issues have been raised. I live in a country where some animals are given a higher status than even human beings. So it's a major issue. I mean, when I was at NIH, I was in the constant threat from animal rights groups. Here also I've received a lot of emails about that. In some media, it's said that why animal has to give his life to save a human life. So those kind of issues do exist. And it also said, like, a pig waited to give his life to save a human or something. And there are a lot of sensitivities about it in this country in particular. Maybe not in Pakistan, but but they issued those statements while eating a steak. So
Yes.
I mean, to be honest, so far, personally, and my team has not faced that kind of that kind of issues yet, but definitely, those are valid issues.
Thank you.
Professor Rizvi, you wanted to make a comment, sir?
Yes, it was excellent. I'm not very ethical, nor I'm intellectual. My question is very simple. I would like what you are doing there to translate it here sooner or later. How to prepare our doctors, technicians to get involved in xenotransplantation, and then later on to human. How can we train them?
Definitely. I mean, and since your focus is on kidneys, I mean, that's that can be done. Liver is a difficult organ. I mean, even here, the survival in experimental model has not gone beyond a month. It can be used as an ex vivo system until the human liver is available. The liver has its own problem of all these coagulation stuff. So liver and lungs may be the last ones to be used in as xenografts. I think the kidneys, islet transplantation, which has already been done in New Zealand, and heart. I mean, these are the few organs which can be, or cornea is being also used. So these are the few organs which I think will be used. But as far as I know, there's no heart transplant program in Pakistan yet, right? I mean, it's not being done, but definitely kidneys is being used, and there's a great demand of kidneys. And the advantage of kidney, as you know, unless all the vessels are compromised, you always have an option of going back to dialysis. So I think, I mean, you know, there is the first of all, even when I established this research program at my current institution, my goal was to not have a separate research team. I involve all the clinical people to get involved. So the perfusionists came from clinical side, the anesthesiologists, cardiac anesthesiologists came from the clinical side, and you know, of course the surgeons and the other staff also, the nursing staff came from. So I had a relative easier time of translation because those people were already aware of the complexities of xenotransplantation when we did this first human experiment, because the same team did the human, which was doing the baboon work. So that is important. That, you know, still now that, you know, it's been done, you know, 104 more people wants to get involved in it, but they don't have the right knowledge. So my advice would be to educate first about what to expect in terms of kidneys also. I mean, you'll soon see that, you know, both at NYU and UAB, they will attempt these transplants with or without FDA permission. Even here, there are provisions that people will try to do few things without permission. So I mean, there is a chance of doing that, but like I said, if we get into the tolerance, then we will require, we could get away with less gene modifications if we can induce thymic tolerance. If that can be applied, that will be an easier start in Pakistan, because then you need maybe like only one gene modification, or if possible, if we can induce tolerance in a wild type pig, which I don't think will be possible. I mean, you know, that you can do those transplants in Pakistan. I mean, I'm here to help. You know, if you are thinking seriously in that term, we can brainstorm, see what is available, what are the opportunities. I mean, I can suggest if we can start on a cellular level, that will be that can also be a possibility. You know, if we develop some kind of relationship here and can transport, like, you know, pancreas, and you can harvest islet cells over there and then transplant. You know, that also requires a little bit work. You know, you will need like 4 pancreas from pigs to transplant in one human. I've done that in my previous life. So I mean, you know, there are several opportunities, and everyone in other countries also are at least asking me the same question. You know, how can we do it here, especially in the third world countries? So I mean, we can discuss that offline, and, you know, see what is available and what can be done.
This is indeed being regarded as a game changer, and inshallah, God willing, we will see you take this further. Now, I think we've taken a lot of your time. It has been a fascinating discussion with you, and we are really, really pleased and happy that you gave SIUT the first opportunity to have you on board and have this very fascinating talk. Thank you very much for your time and efforts, and our invitation is an open one for you to come to SIUT whenever you are able to, and give us a talk in person also. Thank you so much, sir, for your time.
Thank you very much.
On behalf of SIUT, Dr. Prisby, and our team and our panelists, I would like to bring this to an end. Thank you so much.
Thank you very much. Thank you.